Hey, Raag, I noticed that bubble tea is becoming really popular over the last 5 years, and I know that you work on micro bubbles. Is there any relationship there?
No, not really, because we're really studying nano droplets, not microbubbles.
Oh,
Anyway, so hey everyone, my name is Raag Airan. I'm a assistant professor in radiology at Stanford and courtesy in material science and psychiatry. And today I'm going to talk about pharmacologic approaches for ultrasonic neuromodulation. We've heard a lot about different ways about getting at using ultrasound to affect brain activity, and today I'll talk about adding drugs and pharmacologic agents to that mix. And the the idea behind this, or how I got into this, is thinking about this question where you know currently in the most of clinical neurology, psychiatry, neurosurgery, many times we use drugs to treat the condition at hand, whether it's an antidepressant for major depression, an anti-anxiety drug for anxiety disorder, or an antipsychotic for schizophrenia. And that drug will, we know it's effective, but it goes everywhere in the brain and the body with current delivery methods. And whereas in many cases, we have a strong hypothesis that one or a few critical brain regions is maximally mediating the effect, the therapeutic effect of those drugs. And so the action of those drugs at all the areas that aren't those one or few brain regions are yield side effects. And so what ideally you'd want to do is just beam the drug exactly to the one or few brain regions where it'll have its maximal efficacy. And so that's the thing that has been animating my effort in this, and why I've been looking at approaching it this way. And overall, I mean, kind of stepping back, my background for my grad school was in developing new technologies for neuroscience. And I was also kind of thinking at that point, I mean, those technologies were great and highly precise, but also fairly invasive. And so in addition to having a targeted means for using these drug agents in the nervous system, I also wanted this technique to be non-invasive. And so it could have its maximal efficacy, applicability in the clinic. And so if I were to identify like the ideal features of a technique for non-invasive interventions in the nervous system, it'd be able to target any brain region, of course, with a clinically relevant spatial and temporal resolution. And what I would define as a clinically relevant spatial and temporal resolution is a spatial resolution on the order of a few millimeters, because that's the order at which we know that these brain regions change in terms of their anatomic and functional connectivity and so on. As well, and a temporal resolution at least on the order of a few seconds, so that we can, because that's as long as it takes to ask the patient then a question after we do our intervention. So we, if we zap or do something for a few seconds, then we can ask them, well, how did it feel before and after? And they can actually tell us that. Of course, it would have no off-target or permanent non-therapeutic effects. That's kind of the whole reason we're doing this, is to get around the whole idea of side effects. So we don't want to have to prepare or do anything that's going to permanently do something to the tissue otherwise, whether it's damaging it, doing gene therapy on it, or so on. It'd be image guided to account for the anatomic variation that we know exists person to person. So all that MRI guided focus ultrasound stuff that we've heard about comes into play here. So every person listening to this lecture, between the group, there's probably some salient differences in the anatomy of our individuals, just because there's normal variation between human to human, which I see every day that I'm clinically active in neuroradiology. And we would have a robust and predictable underlying mechanism of action. And that mechanism of action should be able to modulate not just one method that the brain uses, the excitatory and inhibitory, but all the different methods that the brain uses for signaling. And excitatory, inhibitory, the varied forms of neuromodulatory inputs. We'd ideally be able to patch into each of those. And I say all this because, especially in this context, because focused ultrasound gets us much of the way there. It can target any brain region transcranially, non-invasively, and meeting that my predefined clinically relevant spatial and temporal resolution. We actually have millimeters and millisecond scale resolution of applying the ultrasound in humans using FDA-approved hardware that we have not too far from me in our clinical, in our hospital. It could also be, it's image guided with either real-time MRI or optical navigation and other techniques that are coming to the fore. You've already gone over some of this, but at least in my head, the beneficial features of ultrasound come down to the physics of this in comparison to other dominant forms of neuromodulation used clinically. So I think, so right now, the dominant form of at least electrical neuromodulation used clinically is TMS, transcranial magnetic stimulation. And on the left here is a false color plot of the current source density of TMS in a human. And you see the full width half max of this field is in the kind of orange color. And you'll see that that is several centimeters wide, spanning several cortical gyri. And what this plot doesn't actually show is that the depth of penetration of this field is only 4 or 5 millimeter at the most. In contrast, this is a post-hoc MRI for a patient that underwent non-invasive, focused ultrasound-based ablation of a region of a small nucleus in the thalamus. So all the way in the very center of the brain. And this was done without having to make a skin incision, without having to do a craniotomy or anything like that, just passing the ultrasound from the skin down. And you see that this spot size here, this little white dot, that's the zone that the ultrasound acted upon focally. And so we can actually focus ultrasound at depth, non-invasively, all the way through to the center of the brain. And why we're able to do that in comparison to electromagnetic modalities in part comes from the drastic attenuation difference of ultrasound versus electromagnetic radiation in tissue. So if we kept wavelength constant at 1.5 millimeter, and focusing is of course a function of the wavelength of the medium of the wave, ultrasound attenuates with about 2 decibels per centimeter, whereas the electromagnetic radiation of a similar wavelength attenuates at 74 decibels per centimeter. This translates into a spot size of clinical brain systems for ultrasound being on the order of a few millimeter, but for electromagnetic systems being like TMS being on the order of more like centimeters. So that's awesome. And just as another way to underline this, this is a brief simulation study that we did. This is actually simulating the use of a transducer meant for uterine fibroid ablations. So not even a brain transducer, but one meant for uterine fibroid ablation. And with that transducer operating at 1 megahertz, so these white boxes are 1 centimeter on edge and overlaid onto a real human, CT examination, the same one that we used in this simulation study. The red squiggle is a full width half max of the ultrasound field. And we see that with that transducer, in principle, we could target almost wherever we want. The amygdala, hippocampus, Broca's area, Wernicke's area, nucleus accumbens, subgenual cingulate, even the brainstem in principle. So we can get wherever we want. We'd have, that with focality that promises no off-target effects, image guided, all fantastic. The only issue is, does ultrasound have a robust and predictable, on its own, have a robust, predictable underlying mechanism of action that could be able to modulate the variety of signaling methods that the brain uses? And I'd say that, Kim is, and folks are getting there. We're definitely getting to the point where we will have that. But right now, we don't have, I feel like there's still some question marks here. And so was thinking about like, what could we, so ultrasound is great from the physics standpoint of being able to focus our energy down to a point that we might want, but what could we maybe pair with the ultrasound that could do it? And then I went back to that other thought I had about the drug delivery side of the question.
Yeah, Raag? Yeah, I think there's no question that there's drugs that people are used to using, and they want to be able to use the drugs that they know about. They're a known quantity. They know exactly what they'll do. So I think there's no question that there's good reasons to do this.
Yeah. So thanks, Kim. And just saying, Kim and I collaborate. We're friends on this. So I'm happy to help. Both. I think we're both happy to help each other to get to the endpoint of better interventions for more precise, intervention in the nervous system.
You can see these two really highlighting the tensions in the field that exist, but still the collaborative nature of this field as well.
Yeah. Yeah. So, I don't see our work at all as, competitive in this regard. It's just all about, getting to be more precise, better tuned, better everything. Yeah. So I mean, the thought was, can we basically utilize ultrasound mediated drug delivery to get to that endpoint? And the idea is that the drug would then mediate the neuromodulation only at the site that was sonicated. You'd have a robust pharmacologic neuromodulatory mechanism that could be easily adapted to the modulation of the variety of brain receptor systems that are out there. The pharma industry has characterized, has libraries of small molecule drugs that specifically, enable agonism, antagonism, or partial agonism at a variety of, specific brain receptors. And so we can capitalize upon that if we had such a platform. In the eventual application, a patient would come in and receive an IV infusion of a carrier particle that contained the drug of interest. The patient would then undergo an image-guided transcranial focused ultrasound procedure of some sort. Should know that this picture is taken from a TMS system. This is kind of what I would envision that we'd want to work towards. They would be doing some sort of psychological or behavioral task or undergoing behavioral therapy while getting the localized pharmacologic infusion in their brain. So the ultrasound would induce drug release from the carrier particles, and then the drug would then be able to act in the brain just at the site to mediate a targeted pharmacologic neuromodulation.
Raag, you might get into this, but are there, if our students are thinking about this, and of course, people in your lab, are there any restrictions on the drugs or molecules people can use or fit inside of the droplets?
So it depends on the system, and I'll talk about, we have one system, and there's other folks working on different systems. In principle, our particles, the one that we make, are able to encapsulate any hydrophobic compound. Now, luckily, or that matches our design kind of thing nicely, because in order to cross the blood-brain barrier, unless you, so there's this thing, the blood-brain barrier, which we've heard about, I'm sure, in other talks. And in order to passively cross the blood-brain barrier, generally a drug has to be small and hydrophobic. And our system is tuned to optimally encapsulate small and hydrophobic drugs. Some drugs do have, take advantage of transporters and stuff like that to get across. And if we wanted to utilize our particles for one of those, then we'd have to change up some of the chemistry a little. We're actually doing that a little bit right now for one project. And we would just have to make it happen. I don't see a severe limitation there, but right now in terms of what we can do more off the shelf is with small hydrophobic drugs. Okay. I included this slide before because I wasn't sure, honestly wasn't sure how much went over this. I'm sure probably Kim has already talked about this, but I just quickly mentioned that you can use ultrasound to focus energy down to a point. And I mean, the main thing for the next slide is that ultrasound can either have thermal effects or mechanical effects in tissue. Where the thermal effects is that you're getting the energy to dissipate locally at that focus to then generate heat. And that heat could then be used for ablation and potentially on its own neuromodulation. And then also on the mechanical side, well, the pressure waves which are ultrasound can compress and expand the tissue, and that can be used to induce cavitation in the tissue, which could itself be used for modulation, or the stresses and strains could be utilized to affect the drug release from the carrier particles. And I know that you probably have already discussed all that, but basically, so there's these thermal effects and the mechanical effects, and also kind of so keep that in mind. The other thing I wanted just to show or talk about, which I don't know if has already been done, is that there's variety of clinical focused ultrasound systems already on market right now that we could potentially utilize our systems for. And I want to just point these out because in designing our system, we kind of are at the mercy of what is already out there or who we could pair with in a way. And so right now, I mean, the dominant player in this field is a company called Psychiatric, which is based in Israel and now in Miami. At least their US headquarters in Miami. And so their system is it's MR guided focused ultrasound systems. They're the best honestly out there. So if we can make our system work with that, that they're, I think, the ones we want to go with. For neuro systems, they have a 220 kilohertz and a 650 kilohertz transducers that are already kind of in clinical activity. And they're designed to be, paired directly with the MRI. And so that's what this is the top image is what the system looks like and the kind of prep and everything that the patient goes through in order to use the system. Another group is called Carthera. They're out in France. And they develop small focused ultrasound transducers that can fit inside a burr hole craniotomy. So it's not quite, questionably, to use the term non-invasive. It's non-invasive to the dura, I guess, but it's still, it's an invasive thing that they have to do to implant this. But the upside is that once that's in there, the patient can get up and walk around and go around with this ultrasound transducer. And in order to access it, you just have to apply the small needle that apparently doesn't hurt to give the electronics to the transducer, so the patient in principle can do it at their house or something with this, and then exactly where your ultrasound field is at that time. Now this is a small aperture single element transducer, so you're not going to get steering. You're going to get this like long field in the brain, but that's something that could work depending upon your application. The anterior cingulate cortex is right along here, so if you put a transducer in right there, you can just zap the cingulate as much as you want.
Raag, I have a quick weird question. Yeah. How much of the skull could you theoretically replace before it becomes a problem? You don't have to give a good answer to that. I'm just wondering if you've thought about it.
So I could see you doing burr holes, a couple burr holes. So if you wanted to line up a couple transducer to target a very specific location or something like that. I don't see doing. We do so-called cranioplasties for patients that have traumatic brain injury and they had to take away a good chunk of their skull for a long time, and then you put in a piece of plastic instead. The thing is, anytime you have any kind of foreign object in there, you have to think about infection. And the larger the thing, the more extensive the thing, the more likely it is that you're eventually going to have infection. So, risk benefit, so for all this. Of course, the minimum that you have to do, the least invasive that you have to do, the better. Okay. And to that regard, so it's not, so the Psychiatric system right now, I think they're working on it, but right now they use a stereotactic frame that is placed and that frame is pinned to the skull. So, you actually have screws that touch or go in by a millimeter into the skull. And so, this has some level of invasiveness too, in comparison to that. So now in contrast, a truly, I would say, non-invasive approach is done. There's a company called Navifus out of Taiwan, and there's other groups, I think, doing something similar. But this is the one I know about. They basically have an ultrasound transducer on a stick. There's a probe on the back, and an infrared camera tracks where that probe is with respect to the human. And so it uses an optical alignment algorithm that is already kind of validated and in clinical practice for other. So there's Neuro-Navigated TMS. Out there. There's also now Neuro-Navigated focused ultrasound. And so this one, there's no pinning, the patient has to hold still on their own, or you put a strap kind of around them in order to help them hold still. And so this is another approach. Now each one of these has pros and cons. I would say the con of this is that you don't actually have then, real-time MRI guidance and the precision that comes with that. You're also not pinning the person, so they can move around, and every single time they move, in principle, would have to redo this registration process. But it is also, I would say, the least kind of invasive to the patient, and because the aperture isn't the whole hemisphere, hemispherical array, you in principle only have to shave if you are shaving, or clip the one section of the scalp that you are going through. It's not the whole head. So. Okay, and I would say there are other groups on the horizon that I know about, and they each have their own kind of way of going about this. Most practical for us, Psychiatric has systems at 250 kilohertz or 220 kilohertz and 650 kilohertz. Carthera system, I believe, are 1 megahertz. Navifus' system is 500 kilohertz. And as we've learned about, all those frequencies matter in terms of things, and that they certainly matter in terms of how we tune our agents, our carrier particles, for that. And then there's also other things about the how long of a pulse their generators and everything can handle. You know, Insightic could do whatever. Carthera in principle could do whatever, but you know, they haven't necessarily enabled it for that. You know, so there's all those things, and also the pressures and intensities that each of these applications, you know, each of these can generate. The Insightic system can go, you know, all the way to ablative things, so there's effectively no limit. Carthera, you know, in principle, it doesn't have as much limit, but the Navifa system might, because it's actually designed for blood-brain barrier opening, and our particles, the encaging happens at a higher pressure than blood-brain barrier opening. And so that's a pending question that I'm twisting people's arms in order to make happen. Okay, that's it. So I'll talk about, you know, and this will be kind of an outline for the rest of the talk. I'll talk about, you know, our system mainly because of course, but I'll touch on other people's systems. But I'll say, you know, I'll show that we've developed ultrasound sensitive nanoparticles that can be used to ultrasonically encage a variety of different drugs. We believe we've established their biocompatibility and safety. We've used visual evoked potentials to measure the dose response and temporal resolution of this technique, and FDG PET imaging to measure the spatial resolution. We've also used FDG PET to map the functional conductivity changes that are induced by uncaging of the anesthetic propofol. Okay. Now, okay, when thinking about this, I mean, and I was, you know, trying to develop this system. Now remember, I mean, so ultrasound can be used for thermal and mechanical bioeffects, and I really wanted a mechanical gating system. And that's because, you know, you could use a thermal gated system, but usually those temperature transition are like at 45 or even 50 degrees Celsius. That 45 or 50 degrees Celsius would heat shock the brain that you're trying to modulate. You know, you could directly neuromodulate the tissue, usually on a negative axis, with that level of heating. Also, from a physical perspective, as Kim can tell you, I mean, it's kind of difficult to heat outside the very center of the brain. So it's basically difficult to specifically heat. I would say, all the, you know, as you get closer to skull surfaces. So for a couple different reasons, the thermal systems aren't ideal.
Yeah, I'm not quite sure what you're referring to. Do you mean like with the hemisphere, it's hard to focus out there? Because you could use just some other transducer that's, you know, smaller, and then, you know, it could focus out there to.
Well, I mean, but you would probably be, my understanding is that the field gets pretty unpredictable as you get closer and closer to the skull surface.
With the hemisphere, it's hard to get outside at the center. Yeah. But you don't necessarily have to use the whole hemisphere like the Insightic system. It could be a smaller system, and then you can focus in right on the cortex.
But then aren't those systems like, I mean, I thought part of the reason of using the hemisphere was also to distribute the thermal dose so that you're not going to like burn the scalp and the skull.
Yeah, that's true. Yeah.
Yeah. So yeah, so if you had like a 10 centimeter aperture, I mean, yeah, you'd probably like be only distributing that into like that much skull, and
yeah. No, yeah, there's a good point there. Yeah.
Anyway, but more so for the heat shock kind of thing was more the thing. I'm like, you know, and I mean, and okay, and so say you designed your system so that's like, you know, gating at like 38 or 39 degrees centigrade. By the way, I mean, 38, 39 is still considered like a bad fever, you know, which has its obvious neuromodulatory, you know, own neuromodulatory effects. But even if you did that from like Boltzmann, we would know that there's be a significant amount of non-specific release of whatever your agent is. So no chemical system I know about will be so like stepwise of a change that, you know, that at 37C it's like 0, and at 39C it's like 100% or 50% even. So all these systems, like necessarily their, you know, temperature of transition would have to be in like the mid 40s or whatever in order to have that specific specificity. And then also practically, you know, you'd probably have to then keep your ultrasound thing on for like quite a while. I mean, like we use what, 20 seconds, or you'd say like 5 seconds, 10 seconds in order to achieve that gating time, in which case, you know, that then inhibits your temporal resolution. Anyway, I wanted a mechanically gated mechanism. So in looking at that, I mean, folks like Natalia Rappaport, a group at, there's a center folks at Michigan, but also in France, that have developed these so-called phase change nanoemulsions, where they loaded them usually with chemotherapeutics. So they were done for oncologic endpoints, where in their systems, what they described is like they'd use like a low boiling point perfluorocarbon, like perfluoropropane, butane, pentane, or even perfluorohexane, and then induce a phase change with ultrasound of that liquid into a gas. And so if that material was at the core of the particle and you load up the particle, then that phase change would be, that liquid to gas transition would be induced with a burst release of the drug. And so, you know, so these nanoemulsions, they existed. And, you know, when I first kind of picked it up, you know, I was like, awesome. And it seemed like it worked, but they kind of had a concerning stability and safety profile, I would say. And so did a lot of work to adapt the existing systems that were there at the time into what I found to be a more stable formulation that would be more applicable for neuromodulatory uncaging. And so we, you know, as a proof of concept, did this. We took the anesthetic Propofol and loaded it into the system. Propofol is a GABA agonist, and so if you dump Propofol onto tissue, it basically shuts down neural activity. And so we were, as a proof of concept, we were able to knock out seizure activity in an actively seizing rat using this technique. Okay, so now I wanted to. So that was where we were at 2017. I just want to briefly touch upon, like, you know, so it's not like we are the only people that do, you know, pharmacologic approach for ultrasound based neuromodulation. And so, you know, leading up to like what I, you know, as you know, I also read, you know, Nathan McDaniel's work at Harvard. So he worked with Marja Livingston to develop a system where they used blood-brain barrier opening to then deliver a neuromodulatory agent. In this case, they used GABA, you know, just the same GABA that GABA channels are you know, open with. And so they did BBB opening, and they systemically delivered good amounts of GABA, and were able to see, they use a sensory evoked potential or visual evoked potentials, and they saw that they reduced the amplitude of those potentials with their GABA infusions. They did that first in rats, and then now Jean-François Aubry and colleagues have kind of validated that this approach could work in non-human primates. So that's good. Upside is, you know, BBB opening is now it's a very well developed technique, and, you know, now in clinical application. And you don't necessarily, you know, you need to, you know, microbubbles, but it's the same microbubbles that are used for contrast, you know, FDA approved contrast agents. And so there's no like exotic fancy particle, whatever out there. You know, in principle, you can do this tomorrow in the clinic if you had a, you know, if it all worked and you had a good reason for it. So, so that's the upside. The downsides, the cons, I would say, are that, you know, this technique has a fairly low temporal resolution because you have to give such high doses, and you have to give fairly high doses of your neuromodulatory agent, because the net distribution of the systemically delivered drug, the net distribution into this volume is actually pretty low. So that most of that drug is still going all over the body because, you know, naked and just going to go there. And because you have such a high like systemic load of the drug, there's relatively low clearance then of the drug from the rest of the body, and so that there's a certain amount constantly infusing into this area. And so that's what you see here. You know, they have a decrease initially, but I should say cut off this time scale. This time scale is minutes, or actually I think like 5 minutes. And so they had to actually like re-up their anesthetic halfway through. So, you know, it's pretty low temporal resolution, which might be, you know, perfectly fine for a given application, but
yeah, we didn't talk about it in detail, the BBB opening, but the BBB stays open for like 4 to 24 hours, depending on how, you know, what parameters you use. And so at a minimum, it's like 4 hours that the BBB is open. And so a systemic release of drug is going to be in there for a while. That's a really good point. Yeah.
So anyway, but it's a good, like, promising thing that, you know, something like this could work. Like, say, if you had a version of GABA that already crossed the blood-brain barrier, which would be Propofol, then you could do something like this without having to modulate the BBB in principle. Okay, and so it was on that kind of like, that being out there that was thinking about developing our system. And so our system uses a liquid perfluorocarbon emulsion, a nano emulsion of a liquid perfluorocarbon. And the drug is loaded into the shell of this liquid perfluorocarbon nanodroplet. So there's an emulsifying agent, a stabilizing agent in that thing. And so the drug, that stabilizing agent keeps the droplet stable, but then also can bind the drug. And then the drug release is induced by ultrasound. And one main upside that we have seen since then is that we've actually kept we've in stabilizing our system, we seem to have lost that phase change. I remember mentioning that the original kind of description of this system was using a phase change. Well, we seem to not have a phase change anymore. And so I think that's actually great that we get our drug release and we don't see a phase change. And so that dynamic huge event means, to me, a safety benefit.
So when you say there's no phase change, you mean it doesn't change to a gas, it doesn't get bigger, it doesn't push onto endothelial cells?
As far as I can tell. I mean, and we've have some data using high-speed optical imaging as well as acoustic backscatter reporting, kind of supporting that a whole lot of nothing happens. I mean, my model is that there's basically an oscillation or some sort of mechanical node excited by ultrasound of the droplet, and then that, minor oscillation, is enough to induce porosity in the shell where the drug is, and that's enough to then cause elution of the drug. We have seen that the spatial and temporal resolution of the technique is basically dictated by our ultrasound parameters on the spatial side and the properties of the drug on the temporal side. And overall, that because we're restricting the volume of distribution of the drug to where we sonicate, we actually need less drug totally systemically delivered in order to achieve the same local effect. So in contrast, in the BBB opening example, you're only distributing drug into these tiny pores, but you have to, since you're giving naked drug, in order to get the same kind of pound for pound action at the site, you have to give pretty high loads to the animal. In our approach, it's actually the other way around. Because we're only releasing the drug, like having active drug where we sonicate, we actually only have to give a little bit of drug to the animal, and it gets a bigger effect at the site of, on the order of a 5 to 10-fold amplification with this method from what we've seen.
I was wondering with the gas bubbles as well, but is there any tendency for these nano droplets to like merge together and to clump into larger bubbles, or is that just something that doesn't happen?
That could absolutely happen. The technical term for that is oswalt ripening or coalescence. And whether we are, with our ultrasound, inducing that is certainly a possibility. And maybe it's part of the mechanism for how we get our release. The only thing is, I mean, the blood is perfusing through that time. So, and we were using like a 10 or say 50 millisecond, 100 millisecond zap. And so it might coalesce for those 50 and 100 milliseconds, but in the 900, 950 milliseconds that is our off time, I would imagine that it would wash away. And so there wouldn't be like an accumulation of those bigger droplets. Also, kind of in our high-speed optical imaging of these droplets, we've been at a resolution on the order, on a micron scale, and our droplets that we use are like 300, 400 nanometer. So if they coalesce, we would quickly be above that micron scale, and we haven't seen that. So it's not saying that doesn't happen, but just we haven't seen it. So I'd say that's probably a minor contribution to what's going on, at least with our system. But great question, of course. Okay. So for our system, we're using FDA-approved constituent ingredients, but we are packaging them into like an exotic package. So we have to do some work to like prove to the FDA for clinical translation, that our package of these FDA-approved constituents aren't themselves like going to do something crazy. Also, I mean, relative to other systems out there, because we're only loading it into the shell of the particle, we have relatively poor drug loading on a per nanoparticle basis, which means that we have to give a lot more nanoparticles for this approach than, say, compared to other systems. So like if the system were to so those thermally gated liposomes, they actually have an oil that you load the thing into the core of the particle. And so the whole core is being used to load the drug, and then the shell is just being modulated to open it up. The perfluorocarbons that we use here, they're great from an ultrasound responsive point of view, but they're kind of like an anti-solvent. They don't dissolve anything. So very few agents out there, there's one in particular that we're working on now that might get around this, but very few agents out there would actually be loaded into the core. So that's one, I think, downside definitely of our approach. Yeah, okay. Now that said, I mean, so we, did a few studies, and nicely, I mean, Clairvaux Hininen also did a similar-ish study where they used perfluorobutane nanodroplets loaded with a barbiturate ingredient to achieve a similar endpoint with a targeted anesthesia of a brain region. And so it's nice to see that, other people can also do this. It's not just our, craziness. One thing I did have a question on that study is that they used a perfluorobutane for their droplet. And we know that at body temperature, perfluorobutane is a gas. So I don't know if it's the same exact system in a sense, because our agents are pretty definitively liquid at baseline and body temperature, whereas I'm pretty sure that these nanodroplets that Clairvaux was using were probably gas, or they'd have to. I would need to see the data that was, yeah. Okay. And then the final system, which actually was just published not too long ago, is from Mehmet Fatihyanek's group at ETH in Zurich, or Switzerland at least, pretty sure Zurich. And so what they did is they made this kind of complex thing where they have a perfluorobutane nano microbubble. And so, yeah, so it's the same perfluorobutane that Clairvaux used, but in this system, it's a bubble. And yeah, so this is what I would expect, that the perfluorobutane would be a bubble. And then they made these nano droplets where the droplet is actually of a solvent or saline to load a known drug into the core. So they have high drug loading per droplet. So they have these nano droplets loaded with the drug, decorated, bound to the outside of a microbubble. So it's this thing. So they basically have this like a linker, a malamide thiol linker here to bind this nano droplet to the shell of the microbubble. And so they have the small molecule agents at the core of the nanoparticle of the thing. So they have these small liposomes on the outside of a lipid coated microbubble. And they say that their system under ultrasound, there's an aggregation, fusion, or maybe collapse of the microbubble that then induces the drug release. They used mice and 2.5 megahertz ultrasound to locally deliver mucimol using this technique. So mucimol is an agent which naturally crosses the blood-brain barrier and has a GABA antagonism as its primary endpoint. So it kind of gets to a similar thing that we were able to see. They saw what they reported very, very high target to background thing, probably because they have good drug loading into the core of these particles. Interestingly, in their paper, they did not, even though they're using a microbubble with ultrasound, that's a decent pressure, they did not see any blood-brain barrier opening, even though there's like a microbubble in there and collapsing, which is the usual mechanism that we use for targeted blood-brain barrier opening. So that's great. As kind of like, so these are all positive points. Things I would have want questions on or what is that, so they have to use this like functionalized lipid coating and all this stuff. And so the question is whether, is that gonna make a challenge for clinical translation eventually? One thing that I definitely see as would be a challenge for clinical translation would be a 2.5 megahertz ultrasound. So you'd have to like modify the system to get probably to lower ultrasound frequencies, or show that it works there. And then there's a couple other things. So mucimol, it's a hydrophilic agent that gets across the brain using specialized transporters. It gets across the blood-brain barrier via transporters. And so how adaptable is the system across things? And then also, are those receptors, are those transporters going to be equivalently homogeneously distributed throughout the brain? So for Propofol, it naturally just passively gets across the blood-brain barrier. Mucimol, I could see a given individual has a heterogeneous expression of the transport that mucimol uses to get through because it's a hydrophilic agent. So I have questions, but they're kind of in the weeds. But it's an interesting system that I would kind of want to play with more. Okay, so back to our system. So our system, we have 3 ingredients that each have at least investigatory FDA approval. There's a biodegradable block copolymer PEG-PLGA, which is itself used in a variety of drug delivery applications, some FDA approved, some not. There's a drug of interest, and right now we're focused on drugs that are already used clinically, that we already kind of know work. And then there's a nanoscale droplet of a perfluorocarbon agent. And before we used relatively lower boiling point agents, and now we're kind of like exploring higher boiling point agents from a safety perspective, and also for looking at agents that are themselves already FDA approved. Under ultrasound, the perfluorocarbon expands or deforms. That expansion and deformation increases the porosity of the polymer layer. This is at least our model. And then that porosity induces drug release into the blood. And notably, in our model, there's no necessary modulation of the blood-brain barrier. So the drug will then, on its own, passively cross the intact blood-brain barrier to get into the brain. We are have validated one method that is a scalable production of sterile and stable nanoparticles using procedures that can adapt relatively easily or scale to pharmaceutical grade production equipment. We have seen that our system is stable through frozen storage and in the immediate post-thaw state. So that so when I was first making these on my own, I would get into the lab at 5 a.m. in order to make a single batch of particles that would be used for like 2 rats. So I could never convince any of my graduate students or postdocs to do anything like that. So we had to make a system that could be like made in a relatively large batch and like used, just take it out of the freezer, inject the, warm it up, inject the animal. So out of necessity and practical utility with that. So that turned out to be a very tough thing, but glad we were able to do that. Or rather, every single student and postdoc in my group is glad we were able to do that. One thing we've also done is we've looked at the effect of the, if we vary the emulsifying agent on our drug loading, and what we see that there's a relative trade-off. So if we take a polymer and if we extend it, if we lengthen it, we have more binding sites for the drug, and so we get higher drug loading. But that polymer is then more rigid. So it's kind of harder then for that deformation process to occur. So we need like a little higher ultrasound energy to get it to do the encaging process. And so there's kind of a trade-off of which polymer you use and everything for that. But the effect of the drug is that essentially if you have a more hydrophobic drug to start off with, you get higher drug loading. But otherwise, it seems that the other properties of the particle system stay the same, whether it's the biodistribution, the uncageability, if you will. So, there's one minor effect in that hydrophobic drugs tend to get out of the particles a little easier, whereas other way around. So yeah, hydrophilic drugs get out of the particle more easily into the medium because the drug will bind the hydrophobic ones more, whereas the hydrophobic drugs then stay inside more. So you there's like a 5 to 10% effect of that. But always like the biodistribution, the clearance kinetics in vivo, and so on, seem to be relatively the same, despite, regardless of which drug you're using. And we've encapsulated anesthetics like Propofol, cardiovascular agents like nicardipine and verapamil, very interesting drug ketamine, which is a psychedelic, anesthetic, and now antidepressant, as well as chemotherapeutics like doxorubicin. We have seen that in our hands, it's, once everything goes right, we don't have any residual solvents and all those things, that as far as we can tell, the process itself is safe, whether we use like clinical measures of just repeated dosing and looking at the animal's body weight over time, MRI measures to see if there's any blood-brain barrier opening, any hemorrhage, any edema, any so on, or histology. We haven't seen at least overt evidence of like a toxic or badness induced in our system. And now, one point is 200, but now a lot of rats have undergone this procedure without overt evidence of toxicity. Again, once we get the nanoparticle formulation right. To measure the dose-response relationship and temporal resolution of the technique, so we use visual evoked potentials while locally delivering the anesthetic Propofol. So Propofol is, again, an anesthetic, so it's a GABA agonist. So if you dumped Propofol into a section of brain, it should shut off that brain region. And I'll note that if you gave just 1 mg per kg of free Propofol to the animal, on this visual evoked potential assay, we don't actually see an effect. You have to go up to 2 mg per kg at least to see that. But in our experiments, we only gave 1 mg per kg. And even with that lower dose, we see that during ultrasound application, after infusion of the particles, we see an attenuation of the visual potential amplitude. And we don't see anything like that with just blank nanoparticles. So this seems to be an effect specific to the propofol and not the ultrasound parameters, the particles themselves, or so on. So again, with ultrasound alone, we don't see any particular effect. And with nanoparticles infused, we don't see any particular effect. But when we combine ultrasound with nanoparticles, we see this attenuation of the amplitude that then recovers as soon as we stop applying the ultrasound. With blank nanoparticles, we don't see a particular effect. If we give propofol-loaded nanoparticles and we apply sub-threshold ultrasound, we don't see an effect. But once we go above our 1 megapascal threshold, we see an effect that then deepens as we turn up the ultrasound intensity.
So Raag, it's not just the drug release, but also maybe radiation force pushing the drug to the side of the vessels and out.
Potentially, yeah. Or I should say, like, our at least with, so with our dose response. So when we have an in vitro assay that kind of like is a phantom or mimic of what we think might be happening in vivo, and so the dose response relationship that we measure, whether with pressure or burst length, kind of roughly matches what we see in vitro. So we do see something that, whether it's a true threshold or an effective threshold, it looks like it's kind of flat before we hit 1 megapascal, and then after 1 megapascal, we see this increase. And so that's what we also see in vivo. So I think the majority thing is that the drug just doesn't get out of the particles until you're past, say, like 1 megapascal, but certainly something else could be going on, like the radiation force stuff too. So yeah, so we see dose response relations with the peak negative pressure as well as the burst length. And then we also, importantly, so if we do this thing in the visual cortex, primary visual cortex, we see our effect. If we move our sonication focus to motor cortex on our visual evoked potential assay, we don't see our effect, as would be predicted. And then when we go back to the visual cortex, we see our effect again. So it's a repeatable thing that seems to be specific to the visual system. And as another kind of thing, so the lateral geniculate nucleus is a key relay nucleus in the optical system, in the visual system. And so when we apply our technique to the lateral geniculate nucleus, we see our effect again. Our effect half-life is on the order of seconds, and that roughly matches what we know of propofol's pharmacokinetics in the system. So this is all in our mind concordant. So now to look at the spatial resolution, we used FDG PET imaging. This is the same metabolic PET imaging that we use clinically, but just a very small scale with a rat. And so this is a metabolic technique. So it's a glucose analog. So this is looking at where the brain is trying to take up glucose. And we see with the blank nanoparticles, we see no particular effect generally across the brain or specific to the ultrasound field. However, when we gave propofol-loaded nanoparticles and applied our protocol, we see this attenuation of signal, so this loss of metabolism in a region that corresponds pretty exactly to where we applied our ultrasound. And that effect deepens when we turn up the amount turn up the intensity on the ultrasound. And importantly, it doesn't seem like it at least immediately bleeds out into the surrounding tissue. Modeling that out, we see that the effect that we see at that sonicated site has it increases with the ultrasound pressure, but not necessarily, but the size of it doesn't increase if we just hold on the timing. What does happen if we increase the timing of each pulse is that the effect size at the sonicated site at the peak actually increases. But if you increase the time, if you increase the intensity of the ultrasound at the site, that doesn't cause an effect at the given site. So what we're measuring is this difference here deepens with the ultrasound burst length, but the ultrasound, the field that the system acts over, doesn't change with just changing the burst length. What does change that is by increasing the ultrasound intensity. That's what broadens out our thing. And so in my head, our model is that once you're above threshold, once you're above that, let's say 1 megapascal-ish effective threshold, all the brain that's captured by, that is above 1 megapascal would be affected. And so as you're increasing ultrasound intensity, you're increasing the amount of brain that is above that 1 megapascal threshold. But in order to increase the effect size, how much of that brain is being affected, by increasing the ultrasound timing, you're basically increasing how much propofol is being infused into that brain region per pulse. And I'm going to wave my hands a lot more and so on, but that's kind of the model that's in my head. Now, one thing should say, so when Jeff very nicely did these studies, he kept, we noticed that these images from the 1.8 megapascal rat were just look dirtier compared to these images. Like, this one looks so nice, and then this one looks just dirty. Because that one is clean as these. I was like, what are you doing? And then he went back and we just laid all 5 or 6 at that time rats out, and what we saw though was that the pattern of the dirtiness or the noise looked similar rat to rat. So we said,
Raag,
okay,
what do you mean by dirty? I mean, what I see is that it's just not as dark in the sort of the whole front half. Is that what you mean?
Yep, and it's kind of heterogeneous.
Yeah, okay.
Right, there's a lot more specks and spots that are out here than compared to here or even here.
It just doesn't have as much FDG uptake in lots of areas.
Right, but that's specific to the sonicated frame.
Yeah.
So the initial images looked kind of like this.
Mm-hmm.
Right. So we're well, actually, is it just noise and dirt, poor experimental technique or something? Or is there something else going on? So to assess that, Jeff nicely co-registered all the animal brains and then did this, basically looked at which voxels were statistically significantly different during sonication compared to otherwise. And this is the same kind of analysis that's done with fMRI or so on, asking that question, kind of the on task versus off task changes in the brain. And what we found is that when you sonicate a chunk of the frontal cortex with blank loaded nanoparticles, nothing happens. These brain regions up here are the Herderian glands. They're basically accessory tear glands that rats have that we don't. And so this is kind of our noise floor for the technique, if you will. If you see changes out there, they're not real. That's kind of like the dead salmon experiment for fMRI. So in comparison with the propofol low nanoparticles, we see kind of a whole network of changes. And so in this false color map, I would say significance is, if we see the dead salmon level of stuff around 5 on the scale, I would take as real anything from green on down. And so we see this whole network of activity changes in the brain. And notably, the brain regions that seem to be affected are known to be sites of functional connectivity to the sonicated chunk of frontal cortex that we were at. So central thalamus, tectum, hippocampus, the olfactory bulbs, is this kind of network of changes. Now, so one hypothesis is that we're just seeing again, heterogeneity and randomness kind of yielding. Now, it shouldn't be random. I mean, these are very highly significant, z-score of -9, I mean, and 8. These are highly, certainly, or it has to be a very unique pattern of stuff to make this happen. And also the fact that, we sonicated here, yet we see stuff on the contralateral brain all the way in the back, and it's kind of this disparate loci of points, which if it's just say, propofol diffusing everywhere, I would expect a blob to just be diffusing through the brain. So, so that's but so we want to do another experiment to kind of establish that this is probably functional connectivity, or to see whether it's truly functional connectivity or just randomness. Now, so one brain region that went down with the frontal cortex anesthesia is the central thalamus. And we know that the central thalamus and the frontal cortex have a bidirectional kind of feed-forward network. And so if you were to anesthetize the frontal cortex, you should see portions of central thalamus go down, and vice versa. If you anesthetize the central thalamus, you should see frontal cortex go down. Also, the central thalamus is notably deeper than the central thalamus cortical region. So this will test a few things. So the prediction is if we anesthetize the central thalamus, we'll see a differing pattern of changes that would include a more substantial region of the frontal cortex in comparison. And sure enough, that's what we saw. So at the central thalamic site, the effect size was similar to our cortical site. So we can go deep with this technique. Good. But
that's really cool, Raag. Could you potentially reverse engineer what you're doing? Sorry to interrupt, by the way, but could you not exactly know where your target site might be and just look at the network and then say, oh, it's probably here? Or is this something that was special to your analysis and what you know about the brain?
I think we would need a better model of functional connectivity in the rat brain, especially. So these rats were under ketamine xylazine anesthesia. So we would need a good solid model of the functional connectivity of, a ketamine xylazine rat brain. But in principle, yes, yeah. I mean, there's definitely some cool stuff. But like, I think, my whole goal in here is not to do so much stuff in rats. I want to, for good clinically important reasons, do this in humans. Like I want to see what happens. Like we know the anterior cingulate is very important for patients with chronic pain, or the amygdala is very involved in patients with PTSD. I want to see the functional connectivity changes that happen if we anesthetize the amygdala, while that's part of maybe a treatment path potentially for the patient. They're undergoing extinction training or so on. And so it's kind of like a broken record or whatever. I'm just trying to get to human, as fast as and ethically and safely as possible. Okay, but yeah, so here, we see that there's effect at the central thalamus, and we see a differing pattern of network changes in these rats, so that the more lateral thalamus and frontal cortex is going down. So just I'll go back a slide. So here we see this pattern of activity, and in these rats we see just a very different pattern of activity, and that seems to cohere with what we understand for what should go down. We at least find citations where these different brain regions are functionally connected or are anatomically connected to that section of the central thalamus that we acted upon. So we were like, cool. Somewhat unexpected, to be honest. Okay. So that was that study. Now, one thing, kind of going back to the phase change, all that stuff. So this is the data I kind of mentioned before. So originally the system was described as phase change nanodroplets. And yet, in all of our things, like we never saw a blood-brain barrier opening. We never saw any like damage that we kind of like should have if there was a phase change. And so kind of wondered about that. And honestly, a collaborator of ours, Charles Caskey, who used to be a student of Kathy Ferrara's, he was asking about that. Like he was like, he couldn't let it go. So we did this thing with Kathy, who's, is now here, fortunately. Where we use high-speed optical imaging and acoustic, backscatter characterization to look at this. So if you look at the acoustic backscatter under sonication, microbubbles will show this like pretty complex, ultraharmonics, subharmonics, etc., around the fundamental frequency. Whereas, and the proper like phase change nanodroplets, if you made those, they also show this kind of complex signature that looks, similar for good reason as microbubbles. The phase change droplets are kind of like proto microbubbles. And if you kept the ultrasound on after you finished converting them, they would then just act like microbubbles. Yet, our nano-emulsions just don't do anything. We have a notch filter here around the fundamental frequency in order to be able to see what's going on. So that's what this is. But basically, there's just flatness here. So that's like one kind of sort of evidence that it's not really the same thing going on. The other thing we did is we did high-speed optical imaging. So now this is optical imaging, so we're limited by diffraction resolution limits here. So our droplets are sub-micron in principle, and so we shouldn't see anything at least at baseline. But if they became microbubbles, then we should see them, because then they would be like 3-4 micron in size in principle. And with free microbubbles, we see this complex like expansion and compression of the microbubble under the ultrasound field until it pops. or cavitates to be specific. But with our nano emulsions, we just don't, again, see anything. So these are two kind of like correlative pieces of evidence that there probably isn't actually a phase change going on. And maybe, and this makes sense, like, we did a lot of work to stabilize our formulation, to be able to enable freeze-thaw stability and all the stuff while maintaining our drug release. And I think in that stabilization process in our formulation, we basically stabilized it to the point that it doesn't do the phase change, which, yeah. So there are some, I guess, cons to that, but overall I take it as just a win. Like, this is from a safety perspective, great. From a stability point of view, great. We don't have to come in at 5 a.m. to make these, all good. Though there are some cons. Okay. So now, after doing that, we're really focused right now on the clinical translation of this system. We are fortunate to be funded by the HEAL Initiative and the BRAIN Initiative to support the clinical translation. And we're targeting 2 different first-in-human trials. Targeted release of Propofol in brain regions that are expected to be giving rise to epilepsy. So there's basically a signature of interictal spiking that occurs with certain patients with treatment-resistant epilepsy, and we'd be using that as our signature to say that if we anesthetize that brain region, then that signature should wipe out. And that will not only validate our technique, but it would also, for the patient, it should validate that that is, yes, the brain region that is most likely giving rise to their epilepsy before they go in for a resection or ablation. It's kind of like simulation of the surgery that they're intended to get. Otherwise, having to do a craniotomy and all that stuff. And also in that process, ask the patient if they can count to 10 and, memorize 7 objects and so on, to make sure that there's nothing else going to happen, or at least predict that. The other trial is to engage that drug ketamine in the anterior cingulate cortex of patients with chronic pain. Ketamine is currently being used off-label to treat patients with chronic pain, but it has side effects. And so the idea is if we bolus infuse that into the anterior cingulate, the hypothesis is that that should be highly effective. That should maximize the efficacy for ketamine for that condition while minimizing the side effects. Now, the process for this, which I'll share just because it's been a learning experience for sure for me, and it's like a monumental task. The first thing, which is I think the rate limiting step really, is to scale up our nanoparticle production to pharmaceutical standards. What we do in the lab doesn't pass muster for what you can actually inject into a human. I don't want to have anyone inject me with the stuff that we make in our lab. And so correspondingly, I'm not going to sign off on injecting anybody else until it meets certain metrics. And we're getting there, but it's a challenge for a variety of not so interesting reasons. But once we do validate that production protocol, we would transfer it to a contract drug manufacturing organization, a CDMO, to do that pharmaceutical grade GMP production. Then we take those particles and validate ourselves their in vitro and in vivo efficacy for ultrasound induced release, but then also their safety. And we have to do that in rats and dogs. And our approach is that we're going to first do that in a limited set of experiments in rat and dogs ourselves for the safety analysis, but then with contract research organizations that are FDA certified for this analysis, we'll do those safety evaluations with them. And then we'll do a brief kind of efficacy evaluation with those contract research organizations using the ultrasound device that we intend to use for the humans to kind of do a walkthrough, if you will, for the experiment, but also to validate that, yes, we should be able to do this safely, et cetera. And the number one concern here is safety. Once we have all that data, the animal data plus the kind of in vitro data, we get an IND, an investigatory new drug application for the nanoparticle, and an IDE, an investigatory device exemption for the device. Even if it's an FDA-approved device, you still have to go and say, well, we're going to use this for this cockamamie thing that this guy at Stanford's trying to do. And the FDA, our IRB wants the FDA to sign off on that, and the FDA also wants us to sign off on it. So we have to get both. We don't necessarily need a combination license, which has its own idiosyncrasies, but we need one for each, the nanoparticle and the device. And once we have that, we go to the IRB. The IRB hopefully says we're good to go, and then we go. And that said, we told the NIH that we'd be ready for the Propofol project in 2023 and the Ketamine project in 2024. And, knock on lots of wood, we are currently on track. Okay, and to describe the Ketamine Project just a little bit, I know it's not related to ultrasound on its face, but the Propofol one kind of makes sense. Okay, you anesthetize the epileptogenic brain region. Well, you should see something to tell you whether that is truly the brain region of interest. So the ketamine one, well, so the anterior cingulate cortex is identified as a critical mediator of chronic pain. If patients are doing an fMRI study, the ACC is one of the brain regions that pops out for pain-related stuff. And there's also preclinical evidence, animal evidence, human evidence, etc. We also know that sub-anesthetic ketamine is currently used off-label to treat patients with chronic pain, although there are all these side effects. So the ACC is activated by pain. Sub-anesthetic ketamine is used to treat pain. And we also see that those same doses of sub-anesthetic ketamine specifically activate the ACC in fMRI and PET studies. So the hypothesis is that it's the ACC that's not just a mediator of pain, but mediator of ketamine's efficacy in chronic pain. And so if you bolus infuse just the ACC with ketamine, you should be able to see some sort of efficacy on these pain-related measures.
Raag, why, I mean, ketamine has a whole bunch of interesting properties, obviously, but why ketamine over some other inhibitory drug that would broadly shut down, brain cells?
Well, so interestingly, so we don't see, and what's not shown in we actually see an activation with fMRI or PET, an increase. So ketamine is an anesthetic, but when it's used in a sub-anesthetic mode, like in sub-anesthetic dosing, it seems to be an activator. Okay. Very waving hands here, but that's basically the sign here is positive, not negative.
But pain also induces activity in this circuit, so it's kind of like maybe it's the temporal coding that you're changing. What's going on here? Am I getting this wrong? I think I'm getting this.
Nope. You are definitely getting it right. It's definitely confusing. We have no idea how this works.
Rob, wave some hands here. We're open to it.
Yeah. Let me show you. There's one more slide I have to. Okay.
Sure.
Oh yes, perfect. Okay. This is very preliminary data. Okay. But this is data of Jeff and David. And Jeff and David are the team of good folks who did that PET study with Propofol. So now what they're doing is an electrophysiologic and pharmacologic characterization of our process with uncaging ketamine in the rat's medial prefrontal cortex, which basically includes part of their ACC, but also some other stuff. So we're centered over kind of the infralimbic and anterior cingulate cortex of the rat is where our region of interest is. Okay, so first note that if you just gave a 1 mg per kg IV bolus of ketamine, you see these pharmacokinetics. There's a rise that happens over a couple minutes that then trails off with a half-life of a few minutes. And this is concordant with other people's literature of ketamine's pharmacokinetics. Okay. And if you gave systemic boluses of ketamine, you see at a low level, what you get is a dramatic increase in gamma band activity. This is with ECOG. And maybe a repression of delta and theta band activity that then also trails off with pharmacokinetics that kind of roughly matches what you see here. And with 5 mg per kg, as you increase the ketamine dose, you see the gamma band activity, but you also start seeing induction of delta and theta band activity. And delta and theta band is like slow wave activity. Okay. So that's cool. And this is basically if you then raise the ketamine dose high enough to get to anesthetic levels, you're going to stop seeing the gamma. You're actually going to repress gamma band activity, and you're going to see more of the delta and theta come up. Okay.
Well, Raag, just for students who aren't familiar with ECOG and myself as well. So when you have the increase of the delta and theta, then that's more like the animal's going to sleep?
So it's a slowing of neural activity.
Yeah, okay.
Now, Keith can certainly tell you that sleep is a very complex thing. So I'm not going to use that terminology. Yeah. But it's a relative slowing of activity. Now, one other thing though I'll note. So the gamma band activity, it kind of goes up and comes down, and I would say that the overall timing of that is not too dissimilar from the pharmacokinetics. Okay? But look at what's happening with the delta and the theta. So the delta goes up, but then it kind of hangs out at the amplitude for a while. Okay. So this is now data with our uncaging thing centered at the medial prefrontal cortex. And this on the right is the individual animal, and this is a group average. And what we see actually, what Jeff sees, what we see, royal we. I see my computer when he sends me the data. Jeff sees in the rat, essentially nothing happening with gamma. But there's a profound like spike of delta and some theta band activity that then persists for a long time, much longer than what the pharmacokinetics imply. It's persistent and it's also phasic. So this is an oscillation, but it's almost like we call it like a meta oscillation because it's a phasic oscillation of the oscillation band. And it's like a 6 to 7 minute period. And we see it repeatedly. Like it doesn't necessarily add up animal to animal, so there's kind of like it gets out of phase and anyway, but it stays persistent across, in this case, 6 animals. So ketamine is doing something that actually I think is like it's almost like a re-entrant circuit or something like that. It locks the state into some sort of, because I mean the other feature of ketamine's action is that a single bolus infusion of ketamine can have action that lasts for days. So it's definitely inducing some sort of plasticity. So yeah, and so I'm going to be incredibly fascinated by what actually happens when you do in vivo imaging and hopefully human imaging to see kind of what trails off. I mean, long story short, we know that it's doing something with ketamine here, but what exactly is going on, I think is going to be the rest of Jeff's thesis. Yeah. Okay. Now the other thing that we are going to try to do, so Tommaso in our group has implemented functional ultrasound imaging. So use the same principle as fMRI, but blood flow change, trying to measure blood flow changes, but in this case with ultrasound, not MRI. And there's a variety of reasons we're doing that. But part of the reason is that this technique is much higher spatial and temporal resolution compared to usual fMRI. So here's a validation that he did, which is like visual cortical. Like the first thing you do in any kind of functional imaging study is the visual cortex, light them up thing with lights. And so he sees that, indeed he could see, not just visual cortex activity, but he actually sees superior colliculus. So he images the superior colliculus, which is like a tiny, tiny, tiny brain region in the rat, but you can see it. So what you do is you take Doppler frame images. So this is power Doppler imaging. And you take that really fast as you can across the whole brain, and then you line that up with your stimulus to see which brain areas are correlated with the stimulus, like in any kind of fMRI study, except in this case with functional ultrasound. Okay, so Tommaso used that technique, validated it, and then he applied it to at least looking at systemic ketamine boluses. And he sees that kind of correlating to the stuff that Jeff saw in terms of the kinetics and everything, there is this induction of activity that stays up for quite a while actually, but then trails off. Not unlike what he sees here. And notably, it seems to correlate more with like what's going on with the delta and the theta maybe. Yeah. And it's actually concentrated in areas like the medial prefrontal cortex. And actually, interestingly, not so much in like, so medial prefrontal cortex, and this here is accumbens. So attenuated relative to the medial prefrontal cortex, but there, as opposed to say the rest of the striatum. So that's cool. So we're definitely right now trying to pair everything together to do the functional ultrasound imaging of the ketamine encaging and so on. The other thing that Tommaso has been working on is he's made a wearable multi-element array so that we can apply focused ultrasound in awake and mobile rats. Because like all these other studies, the animal was either physically restrained or chemically restrained, if you will, under some sort of sedation. And so whether it's the physical restraint or the chemical anesthesia or sedation, that's going to affect all these measures that we're talking about. And so we're trying to move towards being able to do this in this, the awake mobile animal without as much of the confounds of the physical kind of or the chemical. I would say that Jeff's experiments were actually done with animals trained to hold still in his chamber. Tommaso's experiment so far, he has like 0.25 or 0.5% isoflurane on board. So that's definitely going to affect some of this I mean, for sure, but right now, out of necessity, we're using. Okay, so kind of wrap up with the kind of what I hope to enable, and this is kind of definitely far out there, but this is what we're trying to make happen, is a basically personalized precision psychiatry. So be able to modulate a target brain receptor in a target brain region and assess and utilize its role in behavior and circuit physiology so that we can better understand disease as well as predict responses to therapy. So on the diagnostic end, we can do a kind of pharmacologic functional brain mapping to hopefully subtype psychiatric syndromes and maybe predict patient-specific response to regular therapies. On the therapeutic end, seems like we should be able to bolusly infuse an agent locally in a critical target to either sensitize or to be the pharmacotherapy. And on the scientific end, I mean, a lot of these drugs, like ketamine, we don't actually have a great model for how they work, especially in humans. And so, at least I want to be able to learn more about that. And this is a kind of reductionist kind of approach that I think will hopefully be helpful. For specific applications, so for Propofol, I already mentioned that, could this be used for localization of epileptogenic brain regions? Kind of more broadly, can we use this to simulate the effects of a neurosurgery by temporarily anesthetizing a target surgical volume? Could we use this to dampen the pathologic overactivity of certain brain regions as an adjunct to the usual psychiatric therapy? So thinking about the amygdala and PTSD or other anxiety disorders, the anterior cingulate and chronic pain, or the subgenual cingulate and depression. Another thing that I think is fascinating myself is, the potential psychiatric benefit of psychedelics. So psychedelics certainly have other effects than psychiatric benefit. And so the idea is, what if we can maximize the psychiatric therapeutic effect over those side effects by infusing the psychedelic agent into target brain regions? We already talked about ketamine, but there's similar stories out there for ibogaine, MDMA, LSD, psilocin, and so on. I could go on talking about a lot of different reasons, but I don't know, kind of in interest of time, I think I'll just move on. One thing that I want to kind of give a more of a shout out, so it's a work that we've recently done, the work of Muna Arial, who now just left our lab. She's now in UT Austin. So this is the idea that, so there's this thing called the Glymphatic System, which is an exchange process of bulk CSF from surrounding the brain and the ventricles with the interstitial space of the brain. And the long story short is that we kind of hypothesize that we should be able to modulate that with ultrasound, and it seems like we can. Muna we have a Muna submission paper and submission right now, and so please keep an eye out for that. At the least, we should be able to use this for drug delivery, because in principle you can then do intrathecal drug administration, and then use this approach to drive that drug from the CSF into the brain. And these are false color images of that process, where with sham we basically don't see hardly any of the intrathecally administered agent getting into the brain. But with ultrasound, we see a pretty good amount that gets into the brain and then stays there for quite a while. This is just a 10-minute ultrasound procedure done in between these frames and these frames. And then there's a driving in of the thing that then sticks around for a while. And
Raag, is it just your sense that it's sort of radiation force is pushing the drug through the brain?
So the pattern that we see, where it's actually maximal around where the arteries are inputting into the brain, argues against that. It's just a direct mechanical thing. What I think is more likely to happen, so the glymphatic mechanism is that arteries are pulsating, and that pulsating of the arteries is driving a pumping kind of action in the perivascular spaces that then transmits into convection through the interstitium. And I wonder if with ultrasound, we're actually causing that oscillation of the interstitial medium as well as the perivascular spaces to then kind of open them up to allow stuff to draw in.
This has, you already mentioned it with sleep, but this has really good implications for under the theory that sleep opens up this space and causes a clearance of metabolites in the brain. Are you guys, interested in that? I feel like that would be a really cool thing to look at.
Absolutely. So I think, I think we have a thing established here. We have run into, there is also there's like the pro-glymphatic folks out there, and now there's the anti-glymphatic folks out there. So we're trying to convince. Anyway, there's like folks who actually question.
Yeah, I'm definitely aware of the people that are not thinking that's what's happening during sleep, but I'm still on board for the theory.
Yeah. No, so absolutely, I would love to like use like Tommaso's transducer, that thing, and apply it in animals that are running around or whatever, and then see how that affects wakefulness at the time of, and also later on. Or if the animal's already sleeping, how is it doing then versus their weight? Does it affect their overall sleep duration, their architecture of the sleep, etc.
Mm-hmm.
Yeah, would love to go there. Okay, yeah. So that's it. Thanks, and we'll take any more questions.
I had just a quick question. I think we asked most of them during the talk, but I was trying to get a sense of half-life and kind of how someone would use this if they had, let's say, epilepsy or something. And I really love how you're driving everything towards translation. So could someone have a small enough amount of this fluid in like one of those insulin pumps that's sort of secured to their body, or does it need to be a huge bag? Is the half-life of the drug really short or long, or what do you imagine in the future?
I can envision that we make, we figure out how to concentrate the particles down or something like that, so it's not like a huge bag. And this is like, so immediately, in the next N years, it's going to be like you come into the clinic, you get your infusion, you wear your ultrasound network helmet, transducer, whatever thing, get your zapping during the time, so it's going to be very medicalized, like in the clinical thing. But I can envision, like, there's already one company out there where they basically are making what looks like one of those EEG arrays, but it's ultrasound transducers. And they kind of like automatically figure out like the registration problem. So you wear that, so that the person can wear that. And then the thing is just getting these particles IV. So you can actually have like a tunneled catheter that just sticks stably into a port, somewhere. Hook that up and just, you have your bag, I guess, at home. I mean, you can do that. Or I could actually envision, like you said, like those insulin pump things, where you basically have this attached to that port already, and then like you take your capsule out of the freezer, if we can concentrate it enough, which I think we can. Hook it up to the thing, and then the pump goes. And then, you put that on, you wear your helmet, you get your zapping, and then afterwards you take it all off.
That's great. I'm really looking forward to seeing how everything progresses over the years.
Yeah. Certainly. So yeah. So I mean, but I just sent to the lab. There's Hofstadter's law, which is essentially everything will take longer than expected, except even when taking into account Hofstadter's law. So it's a recursive saying adage. And so I mean, the whole clinical like while we are overall with those timelines, we are in progress. Internally I was like, oh yeah, we should be able to do this in just like this long, but it's going to happen. It's just going to take longer than hopefully.
Yeah, I have one quick question for you, Raag. You mentioned what's the frequency dependence? Like, what do you have to change if you're using a different ultrasound frequency?
Yeah, so we think we've seen and we expect that the lower ultrasound frequencies, you need less intensity for the engaging than the higher. And that correlates with the idea that it's a kind of mechanical mode of action. We don't so we've observed that. And there's a couple reasons why, but I imagine that, in line with the idea that it's an oscillation deformation process of the droplet, well, like the residence time at the peaks and the troughs of the ultrasound wave are going to be longer and maybe more effective at drawing it out and compressing it when you have a lower frequency. So we see like cavitation type events are more effective at lower things, but so that oscillation is probably more effective there, is my very hand-wavy explanation. But what we also have seen that frequency dependence drops off faster than, let's say, like mechanical index would suggest. So we might need like 1.5 to 1.8 megapascal at 650 kilohertz, we need like half a megapascal at 250 kilohertz.
Mhm.
So the scaling, you know, it seems like it drops off well. And so so I'd be, you know, totally fine with doing 220 or 220 kilohertz for this, or say 250 kilohertz, or, you know, something in that range for the initial clinical translation. Yeah, because of, you know, we would need less just intensity, less, yeah. Trade-off is less spatial resolution.
Yeah. Okay, that's great. Keith, do you have any other questions? We'll just wrap it up here.
I'm good. Next up.
Okay. Thanks, Raag.